Okay. Okay, can I hear you, Kristina?
Yeah. Can you hear me? Can everyone
I can hear
me?
you. Marco,
I can hear you.
can you hear Kristina?
Yes, I can. Perhaps everyone can mute themselves individually.
Yeah, not everyone is muted, right?
Yeah, not everybody's muted right now. Yeah.
Okay. Thanks, Gordon. Thanks, JB.
This is Sharon. Whoever is admin's iPad is not muted, and I think that's where some of the sound's coming from.
I think that might be me, though.
Okay.
I'm not sure, though, but thanks. Okay, Kristina, why don't you go ahead and get started?
Okay.
Thanks.
Well, good morning, everybody. Thanks for taking the time out of your day to join me. I hope everyone's doing well. As Pat touched on a little bit, this is different than a normal thesis defense. You know, I can't see everyone face to face, but I guess if there's a silver lining to this, it's that we're all in the comfort of our own homes, and maybe some of us are wearing pajamas. That's, you know, there's always silver linings to these sorts of things. But thank you again for joining this morning and for your patience with this. If there's any hiccups or technical problems, let us know so we can try and fix those right away. We're also going to attempt to record this, so if
Christiane just got muted.
Don't know how that happened.
am I unmuted? Can you hear me?
Yeah.
Okay, now everyone else is muted, so that's nice. So my thesis work that I'm presenting today is on the roosting ecology and behavior of the Hawaiian Hoary Bat. My committee members are Dr. Pat Hart, Dr. Adam Pack, and Marcos Gorson. They've really been instrumental in guiding me along in this process, so I just wanted to take a moment to recognize them and thank them. I hope this becomes apparent throughout this presentation, but this was in no way a one-person effort. I'd like to start by acknowledging those who helped make this project possible. First, the U.S. Geological Survey and their support and funding, including the entire BAT project team. Really, without them, this wouldn't have been possible. Also, Sean Moira, he personally collected hours and hours of video data, and just for the greater good of the species, and I'm really thankful that he was helpful in sharing that. Also, some state and private landowners that allowed us access to conduct research on their properties. And finally, the bats have always been a challenging study species, but in many ways they keep it very interesting, and it's worth it. So just a basic outline of how this talk's gonna go. I'll start with some background, just to touch on some brief background and ecology of the Hawaiian Hoary Bat. Then I'll start the first chapter of my thesis, which is the roost selection and roosting ecology. And then on to my second chapter, which is a little more descriptive, but it's the diurnal roosting behavior, specifically during the reproductive season. So for some background, the Hawaiian Hoary Bat is known locally as Hawaiian, which is in reference to its wing shape resembling a canoe sail or a family, as demonstrated here in this picture. This is the only extant native terrestrial mammal in Hawaii. It has historically been recognized as a subspecies of the North American hoary bat. So current genetic research, specifically from Barrett et al. 2015 and 2017, as well as Russell et al. 2015, suggests elevating it to full species status. If everyone could mute themselves, that would be helpful. There's a little bit of background noise. Can you get everyone muted, Pat?
Yeah, the last time I did that, I muted you. It would be great if everyone could just mute.
Can everyone mute themselves, please? Okay, I think that's better. So Hawaiian Hoary Bats are solitary, meaning they don't roost in large colonies. They're also insectivorous, feeding mainly on moths, beetles, stink bugs, termites. This has been demonstrated both visually by inspection of insect fragments with microscopy, studies from Todd et al. 2012 and Bernard and Motts 2016. And more recently by metabarcoding guano pellets demonstrated by Pinzari et al. 2019. They're also tree roosting, but more specifically foliage roosting, whereas some species roost in tree cavities or underneath the bark or are tent making bats. Hawaiian Hoary Bats really roost among the foliage of trees. On Hawaii Island, we've previously demonstrated seasonal activity and acoustic patterns with increased activity in the uplands in the non-reproductive season, which is November to April, and then increased activity in the lowlands in the reproductive season. Although the ecological reasons for upland and lowland patterns are still poorly understood, speculate that it may have something to do with the cloud inversion layer. In the wintertime, the lowlands see an increase in rain activity and perhaps faster moving above this cloud and rain layer for better foraging. Also, females give birth in warmer lowland conditions during the summertime. We've also witnessed short bouts of torpor or like a state of decreased physiological activity, usually marked by reducing body temperature and metabolic rate, which may also contribute to decreased activity in the winter or non-reproductive season. Females give birth to twin pups during the reproductive season, which consists of pregnancy from about April to June, lactation from June to August, and post-lactation and fledging from August to November, with pups fully fledged and independent of their mother by November. The timings of these seasons likely overlap and are probably variable year to year as well as among individuals. But these general patterns have been demonstrated by our own capture records, as well as research by Tomic 1986, Kepler and Scott 1990, Jacobs 94, and Menard 2001. The Hawaiian Hoary Bat has also been listed as endangered under both the Federal Endangered Species Act and Hawaii Endangered Species Law since 1970. And it was designated as the official state land mammal in 2015, as shown here. The main reasons for these listings is lack of information as well as threats to the species. Unknowns include population size, although ongoing genetic research will hopefully help get to this question. Recent work has started to look at distribution on other islands, but the species' full distribution, including seasonal changes among all the islands, is still unknown. Survivorship is another big question. We don't know lifespan, although we can infer from mainland hoary bats that have a lifespan of about 14 years. Adult survivor rates and pup survivorship for the species is unknown. Related to that is also fecundity, although we can look at other again, we still don't know if adult female Hawaiian hoary bats give birth to twin pups every reproductive season, which is probably very, very unlikely, or how common singleton births are. Threats also include collisions with wind turbines, and maybe to a lesser extent vehicles and barbed wire fences, although we know that those still happen. And it's unknown the full impacts of pesticide use and predation, or what level of predation these bats are seeing. And finally, the loss or changes to habitat and what is critical habitat in terms of foraging and roosting. Are they the same? Are they different? Those are big questions. So to get at that habitat question, my first chapter is the roost selection and roosting ecology of the Hawaiian Hoary Bat. To date, there's no published studies directly examining the roost characteristics for Hawaiian Hoary Bat. This information is really important. It can be used to inform development or selection of mitigation areas, and inform potential effects of clearing or harvesting, and perhaps give guidance on key plant species for propagation to benefit bats in restoration and mitigation areas. So again, the objectives for this work were to identify and characterize roost locations, habitat, and tree attributes. Our hypotheses were twofold. One, that Hawaiian Hoary Bats will select roosts with physical features that differentiate these roosts from randomly available trees. And two, more specifically, reproductive female Hawaiian Hoary Bats, particularly those with pups, will select roosts that provide net benefits, including protection from predation and thermoregulation compared to randomly available trees and roosts of non-reproductive and male bats. I'll quickly outline, there was a few levels of methods to go through this. That included our study area, capturing and tracking bats, then obtaining these roost tree and habitat metrics, and then the statistical analyses we did. The study was conducted on the east side of Hawaii Island from May 2018 to December 2019. To get information on Hawaiian Hoary Bat roosts, first you have to track them to their roosting locations. To do this, we had to capture, band, and then radio track them. When we captured bats, we also collected demographic information, age, sex, reproductive status. We also took tissue for DNA analysis and guano samples for dietary analysis. We used unique color-coded forearm bands for identification, and then affixed radio tags to their backs for tracking. Radio tags weighed less than a gram, and the batteries lasted approximately 21 days. Oftentimes bats groom them off before 21 days. As shown in these pictures, this is a typical setup of our triple high net system, which is used often in canopies, taller canopy situations, and it allows us to get our nets into airspace just over 7 meters high. And then on the right is an image of a Hawaiian Hoary Bat in hand. You can see the color-coded forearm bands, which help us identify them in roost trees. And then the radio tag with the antenna is fixed to its back. It's a little bit hard to see, but hopefully you can see it on your screens. For this study, we wanted to make sure that we captured bats in a variety of elevations and habitats. So we chose 8 fixed netting sites, meaning we rotated through these sites 3 times per year. So the orange dots are fixed netting locations, and they included low elevation pasture orchard mixed habitat, low elevation native exotic mixed habitat, and low elevation orchard plantation mixed habitat, 2 mid to high elevation native habitats, and then a high elevation native pasture mix, and a high elevation native habitat. The blue dots are opportunistic netting sites. These we netted opportunistically throughout the year, and these are also in a variety of habitats and elevations. So after capture and releasing the bats, we attempted to track to roost locations. We use both non-directional and directional 3 or 5 element Yagi antenna. Tracking was conducted on the ground from vehicles and by foot during daytime hours using 2 to 4 personnel. Upon detection of the radio signal, GPS points and compass bearings of the signal were recorded. Data were recorded at multiple locations and mapped to narrow down the radio tag bat's location. When we were able to track to a tree or group of trees, we used a variety of methods to try and spot the bat in the tree, including binoculars, scope, thermal imager, We also often stayed after dusk to try and see the bat emerge from a roost tree. The ultimate goal was to confirm the bat at its perch in the roost tree, as shown on the bottom left. That's a bat in a tree, like a hoary bat in a tree. And then finally, when we were successful, we were able to collect roost metrics at these confirmed roost tree locations. So we collected roost metrics at 2 habitat levels, roost tree, and then larger roost stand. If you were able to confirm the bat in the roost tree by spotting it at its perch or through emergence, we collected metrics on that specific roost tree. In some instances, a specific roost tree was not possible to confirm. In these cases, we obtained only broader habitat level, stand level characteristics by using location of a signal or LOAS software. And this helped us derive the estimated XY coordinates of the bat's approximate roost location from the point location and compass bearing during the field tracking. All roost trees identified and confirmed with an associated roost stand, which we define as a 50-meter radius buffer around that roost tree. And then in those instances where roost stands, or excuse me, where roost trees weren't able to be confirmed and only stand level characteristics were available, those were defined as the 50-meter radius buffer around the estimated XY derived from LOAS. So roost tree metrics were obtained by personnel on the ground in the field, and they included tree height, diameter at breast height or DBH, percent canopy cover, and distance to nearest tree. Additionally, if a bat was spotted at its perch in the roost tree, we collected roost perch percent canopy cover, perch aspect, and then also roost perch height. For comparison, we identified 6 trees at random bearings and distances using a random number generator within a 50-meter radius of each roost tree. For each random tree, we recorded distance from roost tree, height, and DBH. For stand level metrics, these were obtained using pictometry software and included habitat classification. We followed the methods and characterization of Jacobi 1989. Mean canopy height was determined by measuring 10 random canopy trees within the defined roost stand using pictometry software height tool. We also measured distance to edge, and that was either from the roost tree or the centroid of the stand when we didn't have the exact tree roost location, and also elevation. And so these 2 pictures here show an example of how that pictometry software works, which is really advantageous for us. It gave us both orthogonal or a bird's eye view as shown on the left, and also oblique views from north, south, east, and west, which allowed us to make those random tree height measurements. Sorry about that. So the statistical analyses we did, we compared tree structural characteristics of height and DBH of roost trees to the randomly sampled trees, with the null hypothesis that there was no difference between roost tree and randomly sampled trees. We first tested the height and DBH variables for normality using the Shapiro-Wilk view test statistic and transformed normal data using log transformations. And did again with the Shapiro-Wilk W test statistic. Then use two sample t-tests to compare transformed data for roost and random trees. Also compared roost perch aspect for roost trees with the null hypothesis that roost perch aspects were uniformly distributed using the Rayleigh's test. For all tests, alpha was set to 0.05. So for our results, there were a total of 125 matting nights from May 2018 to December 2019. And 61 bats were captured and tracked. Broken down by net hours, there's approximately 2,163 net hours, and that comes out to 0.03 bats per net hour. I hope that highlights how difficult these bats are to capture. We were able to track 38 bats to 52 roost stands, and 18 of those bats were tracked to 24 roost trees. Bats were visible at 15 perches within the 24 roost trees. There were some instances where we were able to confirm the bat in the roost tree via emergence, but not confident about the exact location within the tree that the bat emerged from. Therefore, we conservatively were only able to confirm those 15 perches. Multiple roost stands were used by 8 bats, and multiple roost trees were confirmed for 5 bats. One female bat with a pup was not captured or tracked, but was actually located in a tree of a solitary male bat that we had captured and tracked to that tree. There were 23 bats that we were unable to track to any roost level, and additional bats that were captured and not radio tagged during the period of data collection. And 3 maternity roosts were identified in 2019. Also for the tracking events, I'll just highlight tracking these bats. There was 486 tracking events. If you multiply that by 2 personnel and about 8 hours a day, getting to the 52 roosts, that's about 150 personnel hours per roost, which is also an incredible amount of effort to get this information. Just to touch on the maternity roosts we found. So the first was a mother with 1 pup, and that was in an urban habitat in a lychee tree. This was one that we actually didn't capture or track for this tree. She was found in the same tree as a solitary male, although they were in different parts of the tree. We also tagged a mother tracked to a tree with 2 pups, and that was in a mixed non-native plantation habitat, and that she was in a eucalyptus tree. And then the third was a mother with 1 pup, and she was in an agricultural plantation sort of habitat in a paper bark tree. These are the roost locations. The stands are in yellow and the roost trees are in red. Looking at this, it looks like roosts are really concentrated in the lower elevations, but I want to point out that some of that is a function of accessibility. Roads and trail access on the east side of Hawaii Island are much more conducive to tracking in low elevations compared to the upland habitat. So for the roost stands, again, it was 38 bats tracked to 52 roost stands. These range in elevation from 13 to 1,654 meters above sea level. Mean canopy height for the roost stands ranged from 11 to 44 meters, and distance from the centroid or from the roost tree to forest edge ranged from 0.5 to 188 meters. Also, the stand canopy cover classifications, we had 25 that were enclosed or greater than 60% canopy cover, but the next highest was in very scattered canopy covered, which is less than 5% canopy cover. The roost tree species that we identified bats roosting in were ohia, malakia, lychee, mango, ironwood, gunpowder, Australian tune, eucalyptus, and paperbark. It was a wide variety of roost trees. And then for roost tree metrics, again, we had 24 roosts, 22 used by solitary bats and 3 as maternity roosts. If that math doesn't quite add up, again, that's because we had the one roost tree that was used by both a solitary male and a mother-pup duo. Roost trees range in height from 9 to 56 meters. DBH was also a wide range from 14 to 268 centimeters. Percent canopy cover, again, a wide range, 3 to 97% canopy cover. And distance to nearest tree range from 0.5 to 22 meters. There were statistically significant differences between roost tree height compared to the randomly sampled trees. And there was also statistically significant differences between roost tree DBH compared to randomly sampled trees. For the perches, again, we had those 15 roost perches that were confirmed in the 24 roost trees. Perch height ranged from 5 to 24 meters, and canopy cover again was wide ranging from 4 to 98%. And perch aspect ranged from 110 degrees to 334. The roost perch aspect did not differ from uniform distribution. However, if you look at the windrows here, there seems to be some sort of trend. Directional statistics are pretty sensitive to small sample sizes, so perhaps with a larger sample size, we may see results that are significant. And then sample sizes, again, because we only had the 3 maternity roosts, we were unable to do any maternity versus non-maternity roost comparisons, unfortunately. So the main takeaways for this chapter were that Hawaiian hoary bats roost in a variety of tree species and habitat types, including native and non-native. We hypothesized that bats would select roosts with physical features that differentiated from randomly sampled trees, and we did find those statistically significant differences between the roost trees and randomly sampled trees. Moreover, the roost trees were larger in both height, were generally, I should say, larger in both height and DBH than the randomly sampled trees. Unfortunately, because we only had the 3 maternity roosts, we were unable to make those comparisons between maternity and those of males or non-reproductive solitary females. Bats were more readily tracked in the lowland habitats, which consisted of mostly non-native timber plantation, orchard, or urban habitats, compared to native or mixed native habitats that are more prevalent, higher in elevation. So potential roost and upland habitats and native tree species might be underrepresented here. Despite the physical differences in the geometry between these tree species that we found bats in, most bats just anecdotally showed a preference for roosting in the outer peripheries of trees, away from the trunk. And although roost perch aspect wasn't statistically different from uniform distribution, there seemed to be a trend in this preference for the northwest to southeast or leeward sides of roost trees. This might be in response to minimizing wind exposure or also sunlight or thermoregulation factor. They may want a thermal exposure in the afternoons before dusk that could help facilitate rewarming before emerging for nightly foraging. So future research should include tracking females to maternity roosts and then hopefully being able to evaluate the selection compared to males and non-reproductive solitary females. Also focusing more on upland and native habitats might give a better picture of native versus non-native roost tree selection and perhaps seasonal roosting variations. So for my second chapter, which is a little bit more descriptive, excuse me, this is on the diurnal roosting behavior of the Hawaiian hoary bats, specifically during the reproductive season. Information on Hawaiian Hoary Bat behavior has been limited to acoustic surveys or anecdotal accounts, or focused on behavior at wind turbines. But there hasn't been any studies directly examining diurnal roosting behavior for the Hawaiian Hoary Bat. So investigating this kind of behavior at diurnal roosts might give a better understanding of roost function and serve in the development for conservation standards. We obviously know that roosts are places where bats rest during the day, but what are they doing there? Are they vulnerable to predation or disturbance? What are the interactions between mothers and pups? These are important questions that we can really only get at through observation. And again, this information can be used to inform development and selection of mitigation areas, potential effects of roost disturbance, and pup survivorship. Excuse me. So again, our objectives for this part of the study were to identify, describe, and characterize behavior, specifically mothers with pups as well as solitary bats, and really focusing on the reproductive season, which again, pregnancy is about April to June, lactation, June to August, and post-lactation and fledging around August to November. So diurnal roost video monitoring was conducted at 2 separate sites, AMA and DOF in Hilo during the reproductive seasons of 2017 and 2018. Site AMA consisted of 1 lychee tree, and it had 6 perch locations identified within the 1 tree. Site DOF consisted of 3 lychee trees and 1 paradise nut tree, with 10 total perch locations among those 4 trees. Behaviors were grouped into 2 behavioral categories and described according to observed activities. Behaviors were documented in 2 ways. One, the number of behavior, the number of times behavior was observed, as well as the duration or total time spent engaged in that behavior. In previous studies, like Buonacorso et al. 2015, bats were captured, banded, radio tagged, and tracked to these roost locations. In subsequent years, we went back and periodically monitored for bat presence. We used thermal imaging, as you see on the top, as well as spotting with binoculars and scopes to detect bats in these sites in 2017 and 2018. While none of those previously banded bats were observed during our study, other individuals were present, and those are the ones that we monitored. But because these roosts were opportunistically monitored, we didn't capture and track the bats to these locations. We didn't have demographic information for these bats. So for this study, bats were classified as either solitary for a single bat of unknown sex, or maternity when a mother and pup group was identified. Video monitoring was conducted approximately once per week from May 24th to September 14th, 2017, and May 29th to August 28th, 2018. Cameras were set up and recorded roost activity in approximately 15-minute intervals. Video monitoring was conducted in the first 6 to 8 hours after sunrise, mainly due to personnel and weather constraints. And then all video was reviewed in its entirety at normal playback speed. So this is site AMA, which was the 1 lychee tree, and these are the 6 approximate perch locations used during 2017 and 2018 reproductive seasons. This is described as an urban habitat. It's adjacent to some orchards and farmland. This is site DOF, which was 3 lychee trees and 1 paradise nut tree. There was a total of 10 perch locations used during 2017 and 2018 reproductive seasons. This is also an urban outplanted arboretum. So there's two results to this, an ethogram of behavior and then also these diurnal roost activity patterns. So first I'll talk about the ethogram. During 2017 and 2018, there were 11 discrete behaviors that were observed and identified. These behaviors were organized into broad categories, including grooming, excretion, grooming, and social behavior, and then non-categorized, which otherwise weren't classified. This is what that ethogram looks like, but rather than reading this off to you, I think that I will attempt to show you videos of this observed behavior. So hopefully this works. So this is starting with the behavioral category of excretion. And here we have defecate. This is the bat is hanging bipedally or monopedally. Wings are extended from the shoulders with the bent wrists. Body is pushed back away from the wings. And then you can see the fecal palate passing between that wing opening. The context of this is basically to expel fecal waste. Some other species of bat right themselves and hang by their thumbs during urination or defecation. I didn't observe any of that, and this was the typical behavior seen during defecation. So moving on to urinate, again, the same posture we see in defecation, we also see during urination, hanging bipedally or monopedally, wings extended from the shoulders with the wrists bent, bodies pushed back away from the wings, urines passing between the wing openings. The context of this behavior is just to expel urine waste. I didn't see any evidence to suggest scent marking or any other function of this behavior. So moving on to the grooming behavioral category, there were two behaviors that I observed, which were licking and scratching. These were often observed in close sequence, but not always. Vigorous licking motion with the tongue or front of the body, including chest, stomach, wings, genitalia, uropatagium, feet. And then also for the scratching was vigorous clawing or scratching motion with the foot across the head and body. And basically the context for these is self-maintenance and it's a cleaning function. Moving on to the behavioral category of social behavior. Since these are solitary bats, the only social behavior I observed at roost was between mothers and pups. Mother and pup interactions included nursing, grooming, which is licking and scratching, and body adjustments. Often the context of this behavior, excuse me, was, maternal care. In this clip, this is a mother grooming her pups. She does have one climbing on her head a little bit, and there's also another pup in her wings that we probably won't get to see. And this basically was just all interactions between mother and pups. So this is an example of maternal care. This is a mother and pup duo, and this is about a month later. As you can see, the pup is quite a bit larger, and there's only one at this time, and I'll get into that a little bit later. But this pup, you can see sort of rooting around, and then finally it begins to nurse. And the mom seems fairly unbothered by this. I'm sure this happened several times an hour. So moving on to non-categorized, we'll start with hang alert. This is hanging bipedally or monopedally. This is with ear movement, eyes are open and looking around. The context of this behavior is it's an attentive roosting position. the bat is awake, looking around, observing surroundings. In this particular video, you can see the bat kind of, you know, cock its neck a little bit and open its ear, perhaps in response to some sort of auditory stimulus, but its eyes are open, it's alert, it's looking around, it's seeing what's going on. Also similar to hang alert is hang relax. Same hanging posture with the eyes open, except in this case, the context of this behavior is just a standard non-sleeping but resting position. Unlike hang alert, there's not any kind of stimulus that the bat's trying to look at or observe. It's just kind of hanging out there. And then towards the end of this video, you see some subtle movements, but for the most part, this bat is just not asleep, but resting. Now moving on to movement. These are just body movements or adjustments, including rotation at the perch. The context of this behavior is just adjusting or improving the body position on the roost perch. And in this case, the bat is sort of adjusting its foothold on the branch that it's roosting on. Sleep, I think, is pretty self-explanatory. Eyes are closed, wings are folded across the body. The uropatagium, or the tail membrane, is sometimes folded over the wings as it is here. The head and chin are tucked, and sometimes the thumbs or wrists are covering the eyes, as you can see here. This is just the non-active, resting, sleeping position. And then finally, stretch and yawn. Similar to licking and scratching, these were often observed in close sequence, but not always. For stretching, there's the extension of one or both wings from the shoulders and wrists. The body is sometimes tensed for several seconds before relaxation. This is just a deliberate activity. It's usually interrupting sleep or preceding sleep. And then for yawn, it's that slow, wide opening or gaping of the mouth with a inhalation or shorter exhalation of air. And you can see the bat's sleeping here. It sort of, there's a little bit of wind. I think that might stir it a little bit. And then we get the big stretch, yawn, and then it sort of settles back down into a sleep pattern again. So moving on to the roost activity, there was a total of 114 hours, 50 minutes, and 21 seconds of video that was observed and analyzed for the 2017 and 2018 reproductive seasons. Observations of solitary bats accounted for a total of about 86.5 video hours, and maternity roosts accounted for about 20.25 video hours. The results for solitary roost, these were of unknown sex. During the 2017 reproductive season, monitoring sites at DOF, bats used, used 7 perches within 2 roost trees and 1 perch within 1 roost tree at the AMA site. During the 2018 reproductive season, bats used 3 perches within 3 roost trees at the DOF site and 2 perches within the 1 tree at the AMA site. Again, since bats weren't banded, it was difficult to know if we observed more than 1 bat switching among the roost perches between these monitoring periods. However, during the 2017 monitoring period at DOF, there were 4 monitoring periods where 2 bats were monitored simultaneously at different roost perches. And during 2018, there were 2 monitoring periods where 2 bats were monitored simultaneously at different perches. So we know at least two bats were using these areas sometimes. For the maternity roost, in 2017, there were no maternity roosts observed. In 2018, things got a little crazy. During the 2018 reproductive monitoring, a mother with two pups was first observed at site AMA at a perch in late June. I'll note a solitary bat was observed and monitored at that same perch location during May and early June. It's likely that this was the mother prior to giving birth, but I cannot confirm that. So that's speculation at this point. It probably always will be. The mother and pups then moved among two perch locations in July, though we never saw this switching behavior observed on video. At the end of July, the mother and only one pup was located at yet another perch. It's unknown whether the trio was predated upon if one of the pups fledged, although that's unlikely given the timing, or other factors that may have resulted in the change to the family structure. The duo again moved one more time and remained at that final perch until the end of video monitoring for the 2018 reproductive season. So during video monitoring, solitary bats, unsurprisingly, spent 93% of their time sleeping, while the mother at the maternity roost spent about 86% of her time sleeping. Additionally, for solitary bats, 6% of their time was spent engaged in grooming behaviors like licking and scratching, while all other behaviors accounted for less than 2% combined. At the maternity roost, maternal care accounted for 9% of the active time, which the maternal care included the licking and scratching, body adjustments, and nursing, and other behavior that involved both the mother and the pups. And then self-grooming behavior like licking and scratching accounted for 3% of time, while all other behaviors accounted for less than 2% combined. When we look at the number of behaviors that were observed, for solitary bats, the most common behavior was grooming. There was 294 events, or about 43%. Stretching, yawning, and movement accounted for 100 events, or about 15%, 114 and 99 respectively, while defecating and urinating accounted for only 35 events, or about 5%. Hang alert and hang relax were rarely observed for the solitary bats, and that was 32 events, or about 5%. For the maternity roost, maternal care, again, was the most common behavior with 220 events, or about 54%, followed by yawning, stretching, scratching, and licking. While defecation was observed, it was pretty rare. There were only 11 events, or about 3%, and urination at maternity roost was not observed. Similarly, the behaviors of hang relax, hang alert, and movement were pretty rare events, with only 20 events combined, or about 5%. So the take homes for this, behaviors observed in this study were similar to those observed in other bat species. However, again, these Hawaiian hoary bats are solitary, so there was colonial behaviors that I read about in my literature review, like social behaviors in big bat colonies that we just don't observe in Hawaiian hoary bats. The roosts in this particular study were at low elevation, like below 100 meters above sea level. Bats roosting at higher elevations might exhibit additional behaviors in response to lower ambient temperatures, including those short bouts of torpor. So it's worth it to maybe try and investigate that. Additional observations also maybe during the crepuscular time period prior to bats emerging from a roost, as well as these observations in upland habitat and during other seasons might be necessary to gather a full range of conditions and behaviors, and perhaps a more detailed picture of daily activity budgets for Hawaiian hoary bats. Again, it was difficult to know levels of fidelity without having these bats individually identified or with color-coded bands like we did in the first study. Well, it's apparent that, excuse me, there was some amount of roost perch switching within tree switching occurring for both the maternity and solitary bats. We also know there's anecdotal accounts of fidelity to these roost sites because these bats, or bats in general, have been tracked back to these sites, again, in Bonacorso et al. 2015, and then from homeowner personal observation at the AMA site, there's accounts of bats using that particular light tree going back 40 years. So it's pretty evident that at least these particular sites are important for this very small subset of the bat population on Hawaii Island. Genetic analysis, if we were able to capture these, may reveal if these sites are familial trees with young of previous years returning in the reproductive seasons. Lower levels of activity were observed for solitary bats compared to the maternity bat, which is not surprising. Maternal care was mostly comprised of grooming and licking, and then just short body movements that are facilitating nursing. Again, it's difficult to speculate what event might have occurred that resulted in the change to that family structure that we saw in the maternity roost. But I think it's an important and interesting anecdote to point out. Though predation or threat of predation was not observed during our study, it really shouldn't be ruled out at this point. And I think really gathering this additional information and observations at maternity roost will help give a more detailed information and account of pup survivalship until fledging, which is a really critical metric for population viability analysis and has really important management implications. So future research should focus on these maternity roosts and again, expanding these observations into different habitats and other seasons for solitary and maternity roosts. Thank you so much for your time and attention. Again, I want to thank my thesis committee and especially my research team that helped make this possible. I also want to thank my family, especially my parents, for their patience and encouragement, and my husband for his support in all aspects, from being part of my field crew to reviewing my writing and always being one of my biggest fans. And with that, I think we will attempt to take some questions.
Yay.
Thanks.
I can unmute everybody. I haven't done this. I'm going to try and unmute everyone, but the people asking the questions may be able to just unmute themselves too.
Okay.
Can we try that first? Does anyone have questions?
Hi, this is Frank. Can you hear me?
Yeah, I can hear you. Great.
Can Kristina hear me?
I can hear you.
Okay, well, congratulations. Well done.
Sorry, can other people hear Frank and Kristina?
Yes.
Great. Thank you.
Have you found anything in the literature for bats or seen any behaviors where you did have two pups that might have indicated sibling rivalry, where a stronger, larger pup might try to dominate the other, and that might lead to loss of suckling time, or even the mother showing preference to a strong? Do you think that's a possibility for the reason that pup mortality might occur?
So yeah, I definitely think that that could be a factor in that mother pup when it was a trio, I did notice that one pup was quite a bit more active, and I think it was actually highlighted in that first video that I showed. One pup was sort of crawling all over the mother and looked very active, and at that time it wasn't evident in that video, but she did have another pup that was just sort of tucked underneath her wings, and just from my observations was less active. That's only a N of 1, so more observations might reveal that that may be a factor.
Thank you. I'm going to mute.
Okay.
Anyone else have questions?
This is Paul Banco. Can you hear me?
Yes.
Okay, great job, Kristina. I just have a question about how vulnerable you think bats are to predators. If they feel, for example, a rat coming along a branch when they're roosting, do you think they would just detach and fly away, or do you think they're very vulnerable at that point?
I think for, adult bats, they would likely wake up and fly away. I think the big risk is obviously the pups. If they are not volant, then there's nowhere that they can go, and so they're quite vulnerable to predation. I don't know if a mother would stick around during the day and fight off a rat. Probably not. And bats, the pups are left at those roost perches at night, which is another observation period that was not part of the study, but might be really interesting to see what happens at night when the mothers go off to forage and leave their pups at the roost trees. So there's definitely, I think there's some vulnerability, particularly for non-volant pups.
Thank you. Just to follow up, if I may, if the bats that are roosting in fruiting trees, let's say especially lychee, are they likely to be more vulnerable then to rats during the fruiting season?
Perhaps. I think it's really interesting that they're still consistently choosing at least these particular lychee trees to roost in. There might be something special, different about these particular lychee trees. We've also seen people picking or banging on the trees to pick lychee trees while the bats are there, and you'll see the bats they're aware of it. I haven't seen one flushed from the tree yet in response to that sort of disturbance, but I guess it would depend on how close the disturbance is to the roost perch.
Great, thank you.
Thanks, Paul.
Anybody else?
Kristina, this is Springer K. I would just real basic questions. What was the latest date you saw the maternal roost active? And then can you tell me what the tool was you were using for thermal imaging?
So for 2018, the latest date, I believe was August 28th. And that was the end, unfortunately, of the observation period. That 2018 was a interesting summer for a lot of reasons. Those of us on Hawaii Island probably remember that. Yeah, unfortunately, the bat was still there on the 28th of August. Whether it was there or the mother and pup were there after that, I'm not sure. And then the thermal device that we used was a Fluke. The model is TI-450. It's a handheld thermal imager. We have a lot of success with it. It's not inexpensive. I believe that model was close to $10,000. But particularly for the lychee trees or the very densely foliated trees, it was really advantageous for us to be able to spot bats in those trees. The disadvantage is that particularly in Hilo or in the lowlands, those trees warm up really quickly, or if you get backlight, any thermal sort of washes out that picture. So thermal imaging is a really early morning task. And again, I thank my field crew for being on board with that, because there was a lot of very early mornings. And they were troopers. They were great.
Thank you.
Thanks.
Great.
Hi, Kristina. This is Teresa Menard.
Hi, Teresa.
Hi, I have a question.
Sure.
Did you, as lactation progressed from May, June, July, August, did you see the females leaving earlier to feed, perhaps because of increased energetic demands?
So part of a little bit of a hole in this too was that particularly for personnel and then also video monitoring reasons, most of the video was like in the first 6 to 8 hours after sunrise. So we don't have a lot of the afternoon period, particularly when they leave at dusk. A lot of that had to do with backlight coming in the areas that we were videoing. And so we sort of lost a little bit there by not being able to monitor that period. So sorry, I don't have the answer to that one.
Okay, thank you.
Thank
I
you.
have another question. Did you see any changes in the pup coloration going from more red to more hoary?
Oh boy, you know, I don't know that I really paid attention to that, but that's a great question. I might have to go back and re-look at that. They do tend to start off pretty red or darker in color, and then just kind of thinking about the two videos that I showed, they do seem to get more hoary as they get larger and as time goes on.
Thank you.
Thanks, Teresa.
My name is Gideon Ambrosius, and I have a question about predators. So besides rats, are there any other animals, like particularly like introduced predators that are known to be a threat to the bats?
That's a really good question. We have, barn owls, and then also our native owl, the pueo, and then also the Hawaiian hawk, eo, which I would think could predate on bats. I know just from field crew observations, we've seen the Hawaiian hawk in the same area as bats. I've never witnessed, and I am not aware of anyone witnessing direct predation on Hawaiian hoary bats by these, or I wouldn't think it's outside of the realm of possibility though. And I would probably assume rats and cats and other things that can get into trees would be a risk factor for bats, although I've never directly observed it.
Oh yeah, I also want to add that my hometown in Hilo, that live by near Wailuku River, and one time I've seen a bat above the Wailuku bridge there, and I even seen like two fly above my yard.
Awesome. Can I might get your address so I can go hang out in your yard.
Well, but it's only that one time where I've seen, or maybe only twice. I mean, one time at the bridge, the time at the yard, but otherwise I haven't really seen them after that.
Yeah, interesting. Thank you.
But it's like in a Wainaku, by the Wainaku area, that's where it is.
Yeah, we've definitely captured and tracked bats in that area.
Because I saw the one by the bridge, do they also roost under bridges or only trees?
These are, foliage roosting. I've never heard of them roosting in any sort of structures or caves or anything else.
Hi, Kristina. This is Kapua.
Hi.
How are you? Great job. I just wanted to ask you a really quick question about vegetation choice. So I might have missed it, but it looked like you summarized sort of the forest stand characteristics surrounding the roost trees that you identified. But can you speak to the density of foliage and vegetation in the roost trees themselves compared to, because it just seems like lychee and mango are such are such dense trees foliage-wise. Since there's no real patterns with forest stand, it seems, unfortunately, for predicting where they might be roosting, I thought maybe there would be something helpful there about the individual trees.
Yeah, that's also difficult because they roosted in, mango and lychee, which are somewhat similar in structure and density, but then we also found them in eucalyptus and paper bark, and also ironwood, which is totally different than any of those. So that is the real difficulty, I think, in this study, is that they used a lot of different things, and figuring out why they use these particular trees is still a little bit of a challenge. I'm still going back and trying to see if there's something that I can model or tease apart to try and get an answer to that, or is the answer that they're just extreme generalists, which is not extremely helpful.
Oh, I know, they're so difficult. They're just not giving us anything, right?
Right. Yeah. But now that 24 roost trees, I think, was a great start. There's plans to continue and expand on this research. So hopefully we can get more of an answer as we continue.
So I would ask a follow-on question then. The roost height was such a wide range. What was it, 5 to 34 meters? It's really, really tall.
Yeah, are you talking about the within tree?
Yeah, it just seems like, I don't know if there's any indication that your research would push Fish and Wildlife to revisit their recommendation of not cutting trees over 15 feet during the nesting season, since there are some roosting in shorter trees potentially.
Yeah, some of that was a function, right, of the tree itself. Shorter trees, than the perch within that tree is lower. Yeah, that's definitely a tough question. It's food for thought for sure.
Thanks.
Thank you.
Yeah, I'm seeing where the bats been roosting, like mango, lychee, ironwood, and even ohia, and it seems like I guess they seem to like denser canopies because you said, or as on a list where they were roosting, they didn't roost in any koa trees because koa, that they have more lighter canopies. So it looks like the bats seem to prefer more, at least somewhat more denser canopies.
Yeah, I think that's part of it. We did find them on the peripheries of trees, away from the trunk. These guys are not typically roosting, right up close to the trunk. They're on secondary or even tertiary branches, which is always really interesting because you kind of see them flop around in the wind a little bit by being that far out on the edge, whether that has some sort of predation protection, that might be part of it. A big fat rat might not be able to get all the way out on the periphery of those branches. That's one of my theories. I have no evidence to back that up, so but it's just something that I've definitely thought about. Anyone else?
Hi, Kristina. This is Julia. Hi. So with your first study, you mentioned that probably some of the difficulty of getting roosts at high elevation. Did you do any sort of analysis of the number of bats tagged at high elevation and how many roosts we got to there versus the number of bats tagged at low elevation? And
I didn't, but that would be a really easy thing just to point out. Yeah, just off the top of my head. And in talking to you guys, how difficult it is to track in high elevation because there's not a lot of road networks and trails and things like that to get in and get at that bat signal.
Yeah, I think adding that kind of a analysis would really strengthen that point too. Great job.
Thanks.
I also want to add that where I've seen the bats in my place is by Wailuku River. At times when I cross the bridge, you know, there's big swarms of bugs flying. So I think the reason why I saw them there is because they're attracted to the bugs that are there.
Yeah, that's a very likely reason.
I have one more question. At the beginning of your talk, you mentioned that you didn't think a female would have twins from year after year after year. I was wondering why you thought that.
What I meant was I didn't think that, I think that it's unlikely that female bats have twin pups every single year. And that's just because we've caught adult female bats that are not pregnant or lactating or post-lactating in these reproductive seasons. So not all females that we capture that are adults that could have pups, they're not necessarily in reproductive condition to have pups every single year. So I think that's something that we can look at more. There's definitely been a focus for us to look more at females. They tend to be more difficult for us to catch for some reason. We haven't quite figured out why that is. But if we could catch more female bats, that would be great. I think that would help us get at a lot of questions. So that's definitely a focus, to catch more females and get some more of those answers.
Thank you.
Thanks, Teresa.
Any other questions? Okay, well, thank you, everybody. Yeah, she reached almost over 100 people for this, which is a lot for a thesis defense. So this is a good format, I think, moving forward. But yeah, so Kristina and her committee are going to have a meeting in private here, but I just want to thank everyone for showing up and asking great questions. And yeah, I'll just sign out from here, and then we'll continue on with Kristina in her thesis defense meeting.
Thank you.
Great job, Kristina.
Bravo.
Yay.
Job.